All right, so welcome everyone to today's webinar on shedding light on fungal contamination, MALDI-TOF identification services. My name is Sandy Almasu. I'm your moderator for today. So our star for today is Hidayah Labadi. She's a total pro in the world of microbiology and MALDI-TOF tech. Hidayah is a research scientist here at Biolog Lab Services, where she brings her expertise in forensic biology to investigate solutions for customer samples. Hidayah's forever for microbiology shines through in her work, with fungi holding a special place in her scientific heart. She led the charge on the filamentous fungal project, where she conceived and validated our proprietary identification method. Hidayah, thank you for being here with us. Just a couple of reminders. Please keep your mics on mute to avoid any background noise. If you've got any questions or thoughts, please drop them in the chat. We'll dive into the Q&A with Hidayah at the end. So today's plan is very simple. Hidayah will take us through fungal contamination testing using our MALDI-TOF method to decrease processing time and increase the probability of species call. Then we will end the webinar with a quick history of Biolog and our services, then followed by your chance to grill her with your questions. So we're looking for about 20 minutes of awesome content. Here at Biolog, we are a multi-omic microbial identification testing lab. We're ISO 17025 accredited, and we're striving to pour our focus into our customers' needs. So we have a plethora of different services within our scope of genomics, proteotypic, and phenotypic analysis, which allows us to provide a complete polyphasic multi-omic approach to our research services. Today, we will focus on our filamentous fungal protein analysis through MALDI-TOF and how we can get the identification you need with an affordable price. At the end, we will go over these services a little bit further, but for now, I will pass the mic to our expert, Hidayah.
Thank you, Sandy. Hi, everyone. I am Hidayah. I am here to take you through some dirty facts about filamentous fungal. Filamentous fungi is also known as molds. They are a group of fungi characterized by their multicellular structure composed of the long thread-like structures called the hyphae. These hyphae grow and branch out, forming a network called the mycelium. Filamentous fungi are diverse and can be found in a wide range of environments, including the soil, decaying organic matter, and evenly common found in your everyday household. So let's get into how filamentous fungi grows. Filamentous fungi starts their life cycle as spores. These spores are then released into the environment, and then when the conditions are favorable, they germinate to form new fungal colonies. The germinated spore gives rise to the structure called a hyphae, which is a slender thread-like structure composed of one or more cells. The hyphae are basic building blocks of the fungal colony. As the hyphae grows, they branch out and form a network known as the mycelium. The mycelium is a vegetative body of the fungus. It's responsible for absorbing all the nutrients from the environment. The mycelium then extends and penetrates its surroundings, separating the enzymes to break down organic matter. This allows the fungus to absorb nutrients such as the sugars from its environment. Filamentous fungi are heterotrophic, meaning they rely on external sources for the nutrients. When the environmental conditions are favorable and the nutrients are abundant, filamentous fungi may undergo sporulation. So during this process, the specialized structures called the conidiophores develop on the mycelium. These structures produce spores, which are then released to start new fungal colonies. Filamentous fungi can produce both asexually and sexually. Asexual reproduction typically involves the formation of spores through the mitosis, while sexual reproduction involves the fusion of specialized sexual structures and the production of sexual spores. Sexual reproductions lead to genetic diversity within the fungal population. So the growth of filamentous fungi is influenced by various environmental factors, including the temperature, humidity, pH, and availability of nutrients. So different fungal species have specific environment preferences, and these factors can impact the rate of growth and the appearance of the mycelium. So filamentous fungi is literally everywhere around us. You can find it at manufacturing facilities. You can find it within your beer, your pharmaceutical medicine, your crops, soil, and even in your food. So what is the risk of filamentous fungi? Why? So rapid and accurate identification of filamentous fungi is essential to determine product safety. What is product safety? It comes down to recall, superficial infections like skin and nails, invasive infections, allergic responses. And even in the environment, it can contaminate the crops, food, and air, and even cause plant stress. So what makes a fermented fungi idea a challenge? Well, only a very few laboratories have chosen to even apply this technology into their routine mycology workflow. Some of the hesitations may be attributable to the lack of standardization regarding culture methods, extraction methods, databases, and acquisition methods. However, here at Biolog, we love our multi-omic approach to identification, so we can take on any challenge that will help our customers. So we developed a method using a mainstream platform to help you identify fermented fungi to the species level. So the mainstream platform that we utilize in-house is the MALDI-TOF to give our customers a proteotypic analysis. The MALDI-TOF stands for Matrix-Assisted Laser Desorption Ionization Time of Flight. It uses mass spectrometry to analyze proteins within a sample to be able to identify different organisms down to the species level. The best results are obtained within fresh growth. So different incubation times are dependent on the organisms. So we will plate and grow samples that are received to us before running on the instrument. Our quickest turnaround time is our next day analysis, followed by a 2-day turnaround, and then the standard 3-day turnaround. Our fungi samples can grow slower depending on the organisms and are monitored daily to check for sufficient growth, as some may take 3 to 5 days in this phase. So the approach we took, again, is the MALDI-TOF. Why? Because it has a fast turnaround time. It's very cost effective, and it gets down to the species level core. Some of the challenges that the industry may experience while testing different organisms is the sensitive extraction procedure needed for the MALDI-TOF. So why did we develop this proprietary media? Biolog conducted a small study to show how other media do not favor filamentous fungi. As you can see, these 4 universal plates all grew bacteria, so it's easy for bacteria and yeast contaminants. Also, we observed that fermentous fungi penetrates itself into the agar, so it's making it nearly impossible to avoid agar contamination. And that is what led us to our proprietary media. Our media was made selectively for filamentous fungi. It has an acidic environment to favor filamentous fungi and prevent bacteria growth. Antibiotics additive to ensure only filamentous fungi grows. And there's a membrane between the agar and the fungal growth, preventing agar insensitive extraction. The best part, it is made in-house. As you can see, the video on the left shows agar contaminants within the organisms while attempting to extract. However, the video on the right shows how the membrane in the middle enables a clean fungal sampling with no contamination, where the spore is literally moving as slick as butter. And yes, our proprietary media works for so many different fungal samples. As you can see, your organism will sporulate straight on the membrane. Our media ensures no contaminants, is favorable to fungal growth, and speeds up our testing for quicker results that you can depend on. So how do we validate our proprietary media? Our test case validated our ensured the fermented fungi library in our in-house methods using our proprietary media for official use on the Bruker MALDI-TOF system. 9 fungal strains were obtained from ATCC, and the strains were based on their typical customer samples that we get in and covering a range of incubation times. These are our results. As you can see, there are a lot of green dots. However, green is always good, but what does that mean? So green has a high probable species identification, yellow means probable genus identification, and red means non-reliable identification. So here at Biolog, we love the efficiency, so we will test your samples in triplicates to give you the confidence of the species score. As you can see from our test case, all 9 organisms received a high probable species score. So are you ready to start sending your samples in and wondering how? We can receive a lot of different sample types, depending on your needs and workflow. Our permit allows us to receive biosafety level 1 and 2, mixed and pure cultures, liquids and solids, capsules, food, cultured clinical samples, soil, plants, beer, wine, and more. However, on the right, you will see the sample types that currently cannot be submitted. Then, to support your quality, speed, and accuracy for all the services that we offer, our samples that come through Biolog Lab Services for MALDI are tracked using our fully automated LIMS systems. So samples are tracked using barcoding from the time they are entered the lab at reception all the way up to being run on the MALDI instrument. As you can see here, this is a screenshot from our LIMS system to show how we can monitor each sample from the time of reception to completion. We also have a new online portal with a streamlined sample submission process. Here you can see the status of your samples, which include the time of reception, testing status, and when we are to the finish line with the samples, where you can now download your reports. Results are available to you in the portal indefinitely, so you can pull up historical reports at any time. We recommend that if you do have a team who needs access, that you sign up for the portal on their group email so that everyone in your team can submit samples and retrieve reports. My time has come, so thank you all for being a part of our successful webinar. I will now hand over the mic back over to Sandy to give us some insight about our other services and ecosystem before we jump into the Q&A. However, please feel free to start adding your questions to the chat now, where at the end of this webinar, we will be able to answer them all. Thanks again.
Well, thank you so much, Hidayah, for all the dirty facts about fungi and how Biolog Lab Services is available to assist our customers. As you all have seen at the beginning of the webinar, we offer a multi-omic approach to identification. However, we have a full suite of other individual services as well. As our services make an impact across the spectrum of industries, here's what we offer to help you unlock the secrets of microbial identification and characterization. So we do offer a multi-omic identification for all bacteria, yeast, and fungi, as Hidayah explained our MALDI-TOF earlier. But we also have the genotypic approach using Sanger sequencing. We also have our phenotypic approach using the Biolog Odin. We also offer enumeration, total plate counts, total viable counts, bioburden enumeration, visual ID, gram stains, biological indicator testing, water sample testing, purities, and any other special microbiology projects that you're working on. You can reach out to us and we can help you with any method that you have in-house. So one of the hallmarks of our services is their wide-reaching impact. We've been in the business as lab services and for about over 25 years, and for Biolog as a whole, over 40 years. So we are impacting and spanning a diverse array of end users. Our commitment to scientific excellence extends across various industries and sectors, which enable us to contribute to the progress and advancement of the pharma industry, food and beverage, probiotics, breweries, contract labs, animal science research, and many more. So whether you're concerned about contamination or enumeration, our polyphasic, genotypic, proteotypic, and phenotypic testing services can help you reduce costs and guarantee brand safety. So Biolog Lab Services results are all under ISO 17025-2017. We're also FDA registered. We have our USDA permit. We're GDUFA registered, and we're also CGMP compliant. We're open Monday through Friday, 8 a.m. to 5 p.m. Eastern Time, and we're ready to assist you with any live support for submissions and technical inquiries. Our industry-leading standard turnaround time is 3 business days, but if you need it faster, you can choose from same day, next day, 2-day, or even weekend priority services. So as we come to the end of our journey, let me introduce you to our ecosystem here at Biolog. We're on a mission to shed light on the intricate world of microbes and cells. Through cutting-edge technologies and pioneering analysis, we offer fit-for-purpose instruments, a comprehensive database, custom plates, and accredited multi-omic lab services as seen earlier. Think of us as your partners propelling your scientific pursuit forward. So thank you all for joining us on this enlightening exploration, and always remember Biolog's for you. To bring us back to the Q&A session, here's a summary of what Hidayah shared with us. So our Biolog Lab Services here, we can test your filamentous fungal with MALDI-TOF using our method and proprietary media. As we all know, fungal is complex and it's all around us, and it has a sensitive extraction method specifically using for MALDI-TOF. So we use our proprietary media and method to eliminate possible contaminations. BioLog can identify your organisms down to the species level. Our standard turnaround time is 3 business days, but we offer next day and 2 days for our MALDI-TOF services, but it all depends on your organism and the way it grows. We have a portal that is available. You can submit your samples and track your samples from reception to the reporting. So to give you our contact information, for any questions, you can always contact our lab services technical team at labservices@biolog.com, or you can call us directly between 8 a.m. and 5 p.m. Eastern Time at our number shown here. You can reach me at the contact information or Hidayah at what you see here on the screen. So now we can take your questions. I'm going to open the Q&A session, and please feel free to enter your questions in the chat. So Hidayah, first question that we have for you, can BioLog test the fungus in soil?
Yes, we can. We can homogenize your soil and then plate your soil on our proprietary media, and we can go ahead and see what grows.
Thank you, Hidayah. Another question, what is your test turnaround time for Stachybotrys chatrums?
Stachybotrys chatrums. Yes, that is your average black mold. That's actually very common and found in a lot of different places. You'd be surprised. It's probably in your attic right now. However, the standard turnaround time for that is between 48 to 72 hours. So you should be expecting results probably within 3 days turnaround.
Great. Thank you. We have a question also from, if I want to culture fungi from a dry feed additive, which contains numerous fungi spores, how should I operate?
Send them in, dry feed. I'm assuming that the dry feed is additive. It's a part of some type of overall product. So I would send that specific ingredient that you're questioning about and send it in, and let's see what we can plate and grow.
Okay. So what is the lab? Where is your lab located?
Our lab is located in Newark, Delaware.
Okay. If we don't use the online portal, is there another way to submit samples?
Yes, you can obtain a sample submission form from online, and you can submit your samples with that online submission form, and we can go ahead and process it still.
So can you purchase the media?
Unfortunately, this is our proprietary media. So as at the moment, no, you cannot. But you can send your samples in, and we can subculture on our proprietary media for you.
We found black specks on a batch of vitamins during our stability study. Can you test those?
Absolutely.
So what are your steps, Hidayah, for testing?
So for the vitamins, we're gonna again homogenize this. If depending on if they come in a liquid or a capsule, depending on the format of them, we can subculture them to plate them to be able to get them pure and purify, and then to be able to see what grows from when we plate them on our proprietary media.
Okay. Can BioLog test the fungus in soil, which we answered earlier? I'm sorry. This question for me. Can the lab do USP 797 enumeration testing? Yes, we can. We're actually going to be launching our service for USP 797 this week or next week, but you can more than welcome to send your samples now. We are helping a lot of compounding pharmacies by actually validating their methods now and actually getting their staff trained on all the environmental monitoring aspects. But you can definitely send us your plates. We can incubate it, enumerate it, and we can also identify any organisms. Another question that came through for you, Hidayah, can you use Odin for filamentous fungal?
Yes, we can actually.
So Odin, you can actually use it to identify and also characterize filamentous fungi as well. Hello, Gladys. I just seen your, do you have any questions for us? All right, as I promised you guys, it took us 20 minutes to the webinar. Any other questions? I think we answered everything in the chat. If you have any questions, please feel free to add. All right, another question. The feed additive, actually, it's a follow-up from Kyle. The feed additive is called BioWarma, mainly contains deflagrant spores. So definitely, you can send us the feed additive and we can plate it on our proprietary media, grow your organism, and definitely figure out what else is growing, if there's anything. If it's filamentous fungal, definitely there's nothing else that's going to show on the plate but that. However, you can we can definitely consult with you once the plate has grown to figure out which organisms would you like to identify. Do we have any other questions? All right, I'm gonna let you guys go for the day. Thank you so much for joining our webinar, making us successful again. So if you have any questions, make sure you reach out to our lab services team or to myself or Hidayah. We're here to help you and consult with you on any product that you might have questions about. Thank you all and have a great day.